Hi everyone, welcome back from lunch. Now we have Professor Wenying Shou, who is going to talk about, I don't know the title, we will see. Wenying Shou is a professor at the University of College London. She did her undergrad in US, majoring both in biology and mathematics. Did her PhD in Caltech, followed by postdocs at Rockefeller and a couple of other places I don't remember details. But the sort of key feature of Professor Shou's work is combining experiments with mathematical modeling. I was looking at her webpage. She very modestly describes that the mathematical modeling component was introduced by very, very talented lab members. That's very kind and generous of Professor Shou to acknowledge all the students. I'm personally very keen to hear the work of yours. Thank you.
Can you all hear me? Very good. So I want to thank the organizers for the invitation. And I want to thank the previous speakers for setting up the stage for me really, really beautifully. So that saved me a tremendous amount of work. So I want this format to be interactive. So I'll be asking questions and I will see how you answer it. So like we take it kind of sort of like a relaxed fashion, right? Like I have no agenda. We stop whenever we have to, right? So that is sort of like my plan. Basically, I have no plans, right? So let me see, where's my pointer? So we have heard about survival of the fittest, but I want to talk about the survival of the most cooperative. And as we heard from yesterday, Professor Sani's talk, right? So like, let's think of an example where cooperators have this access to this resource, but it cannot use the resource because it's a polymer. Oh, sorry, what happened? I don't know. I pressed the wrong button. Oh, good, it's recovered. So cooperators will pay a cost to synthesize proteases, shown as these yellow triangles. But these proteases must be released into the environment and then chew the protein up into bits, into amino acids, for example. And then some of the amino acids can be utilized by the cell itself, or by the original the original releaser, and some just gets lost in the environment. But sometimes, if there's a neighbor, the neighbor can also take advantage of the amino acids. So then cooperator generates a benefit that can be shared or exchanged. And if that cooperator is lucky, and if the partner is also a cooperator, then it will also make proteases, which will degrade proteins more, and then both would benefit. So now I come to the third criterion. So as a cooperator should gain a net benefit if reciprocated. So we have homotypic cooperation. I meant cooperation between individuals of similar phenotypes. And then we have heterotypic cooperation, as seen mutualism, where two different, say, species would provide each other with a benefit than the partner requires. So then both are cooperators if they both pay a cost, but for the purpose of the discussion, I will call the cooperator, the focal cooperator, as cooperator, and the other I just call partner, even though the other one is also a cooperator. And we call this heterotypic cooperation, which has been observed in mutualism, in many forms of mutualisms. So, but in both cases, there can be cheaters, right? So the cheaters, they pay little cost, they can pay less cost, and as a result, they supply a little benefit, but they exploit the benefit. So then they can be fitter than cooperators. So that is my definition of cooperators and cheaters. So then cheaters will increase in frequency because they are fitter. So how might cooperation survive cheaters? So I want the audience to tell me what you have learned over the past 1.5 weeks. What are the mechanisms that can stabilize cooperators against cheaters, despite cheater advantage of cooperators? Can you say it slowly?
So if cooperators can stick together.
Very good. Stick together, right? Yeah, and any other? It's sort of like forming a spatially structured environment if they stick together, rather than randomly interacting with everybody else, right? Yeah, another one, yes. Very good. Recognition. You guys are excellent. Absolutely excellent. So that's exactly what I'm going to say next slide. So there are two mechanisms, like roughly speaking, two classes of mechanisms, right? So this is recognition, right? So where cooperators selectively interact with other cooperators and exclude cheaters. And the other is spatially structured environments. So for example, if cooperators and cheaters, they are in like on some petri dish, for example, the cooperators will give birth to cooperators. So they're surrounded by cooperator neighbors and cheaters too. So that way, the cheaters cannot really take advantage of the cooperators. So cooperators now can increase in frequency, despite cheaters could have grown faster if cheaters were mixed with cooperator. But then the question becomes a little surprising, right? For henotypic cooperation, in a spatially structured environment, you would imagine all these types of cells will again cluster with themselves. But what the cooperators need are not its own benefit, but it's a benefit from the partner, right? And then how do you resolve this? But experiments, right, from Will Harcombe's lab showed that just indeed, the spatially structured environment would indeed stabilize heterotypic cooperation. So we want to understand why. Okay, so okay. So engineers, so I want to introduce to you my favorite model organism, which is engineered yeast cooperation cheating system. So budding yeast, so I'm working with budding yeast, Saccharomyces cerevisiae, which you use to make your bread and brew your beer, right? So a very friendly organism. So I knock out a gene that's required to make lysine, which is amino acid that's essential for the cells. If you don't have lysine, you're dead, basically, right? So the normal yeast can just make their lysine perfectly fine, but we deleted one of the genes required to make this. So as a result, they become oxotrophic. That's the biologist's jargon about to say that these cells cannot, right? They require externally supplied lysine to survive. And then I make them overproduce adenine. So adenine is a nucleotide, right, which you have to use to make ATP, to make DNA, RNA, so on and so forth. So it's like a base, it's a nucleic base. So cells normally would just make adenine for itself. It's very smart, right? If you're making a little bit too much, what it will do is that adenine will go back and inhibit the first enzyme of adenine biosynthesis. So basically, they don't ever make unnecessary amount of metabolites for themselves. So it's called end product feedback inhibition control by the cells. But previous, like decades ago, geneticists studied these mutants, and they found these called feedback-resistant mutants. So these mutants, they just keep pumping out, they keep synthesizing adenine, and they are no longer sensitive to the end product inhibition anymore. So as a result, they overproduce, and I call this L minus A plus, because they cannot make lysine and they overproduce adenine. And the partner strain cannot make adenine or overproduce lysine. It's exactly the opposite. And then the overproduced metabolites are released into the medium, right? And so we call this cosmorphic cooperation. That's a synthetic and mutually obligatory. We can also make cheaters. So cheaters cannot make lysine because we messed with their lysine biosynthesis. And then they just make normal amounts of adenine for itself, right? It doesn't overproduce, so it does not contribute any adenine to the partner, to the green cells, which needs adenine. So it just takes lysine, and all three strains are non-mating, in the sense they're same mating type, and they are further inhibited from like mating type switch and mating. So they're completely sterile. So you can think of them as reproductively isolated. So you can view them as different species if you wish. They're also differentially fluorescent. So one's red, one's green. I actually hope there's nobody colorblind here. But anyhow, so I'm sorry if this, I will point out as I say. So one's red, one's green, and the cheater is blue. So they're differentially fluorescent. So now so what do I have to do to convince myself it's a system of cooperation cheating? Right? Given the definition I just gave you, like, if I say it's a system of cooperation cheating, would you just believe me? Yeah, that's a very good question, right? Because that's a requirement. It had to be cost. So how would you check whether there's a cost of overproducing metabolites? Does your growth rate slow down because you're overproducing, right? That's very good. So, but then how would you measure that growth rate? What did you say?
If they're sterile, I don't know if they'll even grow, right? So are you saying that if they're not mating?
That's very, very good. So, so now the question is that whether, so we want to see whether they have cost or not. So we want to measure their growth rate. But if you don't provide the lysine, they won't grow, right? So then how would you measure? So like then I can say, well, just like physicists is doing approximation, I can do approximation too. So I'll grow them in rich medium full of lysine. And then what do you do? Okay, so that is definitely a valid experimental design, except it won't really work in real life. The reason that the fitness difference is so tiny, it's like 2-3%. But from evolutionary point of view, it's huge, it's enormous difference. But experimentally, just your test tubes, put it in the incubator, the temperature fluctuations of this is already enough to give you enough noise, so you cannot distinguish this. So biologists thought of a way of getting around this problem, right? How would you get around this problem? Right, it's like tiny fitness difference, right? You want to measure that. I don't really expect the answer, right? But I just want you to think about it because it's really clever for some biologists to figure out how to do that. But I will wait for a couple more minutes. You're almost there, almost there. It's 50% there. There's still like missing 50%. But what do you mean by running it for a few generations? Yeah, but the things that I want to say is the environmental fluctuation, right? Already messed you up because like, the temperature, like we have very nice incubators, but every time you open and close, right, the temperature will change a little bit and the cells are very sensitive to temperature changes. You can almost use growth rate as a thermometer. It's that good, right? So then, you don't know whether the draft comes this way or that way, right? And very much like you will have pretty high standard deviation that you will not be able to tell the difference. Very good. It's called competition experiment. Competition, right? So you mark one with one color, the other with the other color, or you mark with barcodes, whatever you want, in the same tube, right? And you look at how ratio changes over time, right? If the ratio, so what is the scenario that you will say this is a cheater? Like if we mix the blue and the red, right? The L minus cheater with the red, L minus A plus cooperator, right? And then you do that. In what scenario would you say, this is indeed a cheater? In frequency, correct. That's exact. And we measured it. Indeed, they're like 2-3%. And of course, to be rigorous, you have to swap color. You want to make sure it's not due to different fluorescent colors, and we did that too, right? So that you're assured that its fitness difference is not due to the way you assay it, but rather is genuine due to metabolite overproduction, which is not surprising. Because if you look across evolution strains, they all have this mechanism, the end product inhibition control. So it's not there for nothing. I don't believe, generally speaking, if you see a feature showing up again, it's very unlikely it's due to chance, right? Because that's a definition of not being a chance, right? So like, okay, so now why do I engineer such a system? Why do you think, what might be the motivation? Because there are a lot of problems with engineering a system, right? And so yeah, in fact, that's what I should ask. What are the pros and cons of engineering a system? Very good. Anything, anybody want to add something to that? Yeah, I think this is a very good answer, right? So first of all, because it's engineered, I can eliminate all possibilities of partner recognition because they've never been cooperators, right? They couldn't possibly recognize each other. Well, sorry, like it's again. The second, you can just like what you say, you can control the environment. You can turn cooperation treaty on and off by controlling what's in the environment. And third, because it's engineered, so I know exactly which interactions we should track. So then I would know what to quantify, right? And then we can, because it's quantifiable, then we can construct predictive mathematical models. And then there's, so then in the models, actually, what do you think are the use of math in biology? I'm just curious, right? What do the audience think about that? Yeah. I would say chance. Just happens to be the yeast geneticists made overproducing mutants for those. So I just chose those because they're available. So I don't have to figure out what the mutants are. So I just wrote to them. Actually, I was very lucky because they were retiring. Because while I was, when I was doing the experiment, I realized it was just at the cusp of retiring. Some have retired, but they still had this freezer. So I wrote to them and said, please send me your strains. So it did. And whoever sent the strains became, became Cosmo. But Andrew Murray also later did like a leucine tryptophan, kind of like a cross-feeding kind of system. So it's not the only way of doing it, just like largely by chance. Yeah. So what is your view? Because many of you here are like physicists, mathematicians, some of you are biologists. Like, what are your views, right, of the use of math in biology? Yeah, it's be able to quantify. That's very good. But I don't want just a couple speakers, right? A couple enthusiastic students dominate, right? The whole conversation, right? I want some other people say something, like other students. Yes, please. I will defend the biologists here, right? Because I will defend it. Because you see, you're right. It's a lot of observations, right? But so I can say like a model. So it goes more philosophical now. What is a model? Model doesn't have to be a mathematical model. It can be a verbal model, right? And so geneticists have done a lot of mechanisms, right? So they say, well, what kind of mutations can I introduce such that the event doesn't happen? So then I know, well, this gene, if this gene is required, it must act somewhere in a pathway leading to that phenotype, leading to that phenomenon. So actually they know the mechanisms. So in fact, they can give you like have you seen this huge pathway diagrams, right? Of how the signal is perceived, how it travels all the way to nucleus, turn on some gene, something come out. This is a model, like, so you cannot say they just observe, right? And I think biologists will kill you if they hear you saying that. But it is right, it's generally not quantitative, right? Partly because it's so complex, right? It is very complex. So again, I'm defending them, right? So it is very complex. As some of which you will see here, right? Because I have a very, very simple system, right? And you will see, the complexity of biology will, like unveil itself gradually throughout my talk. And anybody else, right? Like, so I'm curious, right? Like what, yes, go on. That's right. It's very hard to intuit on anymore, right? Like if it's a feedback, like what would happen if this happens, this and that? Like exactly, they're very, very good. Any other?
When you write a mathematical formula about a system, it sort of compresses the description in a one tiny little thing, and then it sort of describes everything about the system.
That's very good. It forces us to be rigorous about what we're assuming. And exactly, it's very good. I didn't list quite as many as the productive audience here, but in my class I did, but not for this one, right? Whether to verify whether these assumptions can generate. If it generates, does not mean it's true. It just means it's not outlandish. It's not right. It's good if your assumptions in the model cannot generate experimental results. That means something is wrong. Either your model is wrong, or your experiments is wrong, or both can be wrong, of course. And the disagreements between this, that means important pieces are missing, just as I said. And you can, just as one of you said, you can explore variations, right? Because some experiments are really hard to do, right? But with a model that captures the qualitative features of some phenomenon, you can ask, well, what if I vary, say, diffusion rate, right? Diffusion constant by like 1,000-fold, which is experimentally very, very hard to do. Then you can see, oh, what happens, right? So it gives you intuition. So, okay, so we go back to Cosmo, very good. So I just want to go through, right? Like the equations are very simple. So first I want to go through top section with stated variables, right? So there are two populations, L minus A plus and A minus L plus. There are two stated variables to describe, release the metabolites, right? It's like concentration of lysine and adenine in the medium, right? So I have a bunch of parameters. So I want somebody to explain to me equation one. What does equation one say? Speak louder and slower. Yes. Yes, the birth minus death. And the birth rate depends on lysine concentration, because these cells need lysine. The second one is similar, so I'm not going to go through. So somebody, what's the third one? Somebody explain to me the third equation. Yeah, that one. And all the parameters, like the meaning are there. Not you, because you have said so many times, but thank you. Yes, that's it. So RL, if you look up there, did I say something? Oh, I didn't. That's R. See, it's italic with a little, sorry, with a little, so R is a release rate. Yes. That's right. That's right. Yeah. Consumption, exactly. Yeah. So the birth, right? Each birth, it's BL this, right? Each birth consumes that much of lysine, right? Yeah, right. Yeah. So what are the assumptions in the model? Because you said, right, because it's very condensed, you can immediately see what assumptions I made. What assumptions did I make? What did you say? The oh, it's RL, our release. R means, you mean this? Yes. Oh, so the birth rate, so it's this the birth, right? So it's like these amounts, it's just copied from up there. It's copied, do I have the pointer? It's copied from this term, right? This term, the birth. Each birth would consume, you know, like some amount of lysine, right? Because birth, they don't just give, you know, give birth to new cells without consuming, right? They have to consume a certain amount of lysine in order to generate a new cell. And what are the assumptions here? That's right, that's right, that's right. I'm also assuming that no other nutrients are limiting, right? And very good, very good. Okay, good. And also it's well mixed, right? So yes, okay, so very good. So how do I measure, like how would I measure, you know, dynamics? So what we do is that we, you know, because we don't, because of course you can, what are the ways you know of measuring cell density, cell numbers? What are the ways you know how would you do it? OD, but see, the problem with OD is it's a turbidity. It's a mixture of two, because I want to know the red versus green. Plate, exactly, you can plate, but it's tedious, right? You have to pour plates, you have to pour it, you have to like plate it, you have to wait, so it's late, it's delayed, and then you have to count, which is not exactly fun to do, right? So like there's a very clever way of doing that. So basically you take a sample, you mix it with some fluorescent beads, standards, and you have counted, like we have counted the density of the beads by looking on the hemocytometer, for example, or Coulter counter. You can count the number of, you know, the absolute amount, the absolute numbers of beads per like unit volume. So that is known. And a death dye, right? Because we want to also know which is alive, which is dead. So the death dye basically is a dye that, you know, it's like DNA binding dye. And if your cell is intact, it won't get in, so it would look dark. But if you're kind of dead, or if you are dead, then your membrane is kind of like compromised. So the dye will get in, get to the nucleus and stain the DNA. It will shine bright red. And then we have a machine called the flow cytometer. So what flow cytometer does is really, really fun, right? It's invented by physicists, I believe. So basically you have like fluid, called fluid focusing. So it's like laminar flow kind of thing. And you basically focus to a very thin stream. So the cells have to march in single profile, a single file, right? It's just like they're focused. So then they march through lasers, right? Laser 1, laser 2, laser 3, laser 4. And then each, accompanying with each laser, there will be multiple like PMT, photo multiplying cubes. So if emission, right? So depending on the filter, if it's red, like maybe this one will shine up and say, oh, that's event. But if I go to green, I will not register any. If I go to blue, I will not register any, right? So it can do thousands of cells. It can count thousands of events per second, right? So then you get the data right away, and you don't have to plate and so on and so forth, right? So this is one of the data, and there are many channels. So the X, Y axis, just something kind of almost like randomly, it's like some fluorescent channels, which is not important. The bead is really, really, really bright. That's why this is bead, right? So like these are beads, and you can see single bead, double bead, triple bead, and you can see clusters, right? So because you know the percentage now. Like these are beads, these are non-beads. So then you focus on these, you gauge them for forward and side scattering. And you can see all these are like the dusts, because we know these are dusts because if we look at whether they're fluorescent, they're not fluorescent. And because we know our cells are fluorescent. So then we focus on these dust things. And then we, again, check with the death dye. So if it's really bright, that means it's dead. And if it's not bright, it means it's alive, right? And then out of the live population, we gauge again, ask which is red, which is green. So now we know the relative, we know fractions of each cell type. And then we know the beads, right? We know the ratio of the cell to beads, and then we know the bead concentration. So we must be able to calculate cell concentration. And that's how we get population dynamics or community dynamics, which looks like this, right? So in this experiment, what we did is we mixed the two types of cells at 1,000 to 1, 1 to 1, and 1 to 1,000. And we, like propagate. We propagate it like, at some, you can see it came down because we did dilutions, because we wait for the turbidity to go up. But we don't want glucose and so on and so forth to be limiting. So then we dilute down and you see this. But you see the ratio, regardless of your starting conditions, they always go to like this steady, level. It's about 1. Why do you think if I start at different ratios, I would get to around, like the same kind of ratio? The ratio of the two. Sorry, yeah, the ratio of the two. So I can start with many different ratios, right? Like very extreme ratios. So this was 1 to 1. This was extremely enriched for this first, and that one extremely enriched for that one first. But regardless of what I start, I just do, to propagate enough, hours. Eventually, they will come down, right? The ratio will converge to the steady state value. Yeah. Can you elaborate on this? That's right. So it would need to reflect, right? The supply, it's like a supply consumption balance, right? And I want you to think of extreme case, right? If there are many, like, if they're like, so for example, if like, say I'm, so I'm releasing metabolites for these chairs. There are many of these chairs, right? So these chairs all give me something, and I have very few of me. So I get a lot of benefits, right? I'm not competing for this, so I'll grow fast. But the chairs are competing for the very little benefit I give them, so they're forced to slow down, wait until I catch up. And the opposite. So that's the intuition behind that. And you can mathematically also show the ratio depends on the strain ratio depends on the ratio of supply and consumption. But if we look at the total, total population, total community size growth, so initially there's a lag phase, right? So there's a lag phase, and then they grow because the ratio is fixed, right? At this point, the ratio is fixed. So then it must grow at the same rate, right? Otherwise, there's no chance the ratio will be fixed. So that's why they grow exactly the same way. So they're parallel. The green and the red are parallel, and which is the totals, of course. They all should have exactly the same community growth rate as the community growth rate. So is everyone with me? Any questions at this point? What did you say? Oh, add the beads for the facts. Yeah, exactly. We go from from ratio, right? How many percent is this? How many percent that? But maybe when I sample it, this one has more. I don't want a relative abundance. I want absolute abundance. Yeah. We add beads because we count beads because I don't want to count every sample, right? Because that would drive me crazy completely, right? So but then if I add beads, I just added this stock of beads, which we carefully count many times to reduce the error, to reduce the measurement error, right? And then we use that as standard to do this. Any other questions? Yes, Sue. Very good question. They're not symmetric. They're not symmetric in the sense that, just because of the choice of these strings. So I don't quite understand why, right? But the adenine minor cells, they tend to be very reluctant in releasing lysine. So they will wait, and not release it. And then they start, when they're sufficiently starved, they start to release. And the lys minus A plus cells is a biology complexity, right? So because I didn't expect this, right? So like, why this asymmetry? So the L minus A plus will release immediately almost, right? So like the minute they are depleted of lysine, they start to release. So I don't know why this asymmetry, that's a very good point. That causes this, these kind of like abnormal behavior, like at the beginning. So yes. Yes. Yeah, I didn't. So here I did not add cheaters. It's just the two cooperators. I've not added cheaters here. I just want to see what's going on. So the first question is whether they would even survive, right? It's not guaranteed. If I had Cosmo, they will necessarily survive. Actually, that's a very good question. What are the things that, if I said I made some string L minus A plus, A minus L plus, what are the possible outcomes that will make it not survive? Yeah, like, do you think every single thing I construct will survive if I just, played with A minus, or like played with adenine and lysine over protection? Is it a guarantee that that would work?
No, because you could have cases where, as you said, sometimes it takes very long for your strain to generate that thing. So
Very good.
by that time, the other population dies.
Yeah, that's very, very good, right? That's one thing, because even if you release, but if you just keep being reluctantly releasing, even though eventually you would release, it's too late by that point, right? That's a very good point. And then what are the other possibilities, right? So that's the one failure mode.
It was not obvious. So when you started talking about this, it's not obvious that these cells would have the transporters required to get this from the outside into the cell from the external.
That's a good point. That's a very good point. But so it is true, like the overproduce does not mean release. That's a second point. Yeah, but.
Or if it's released, but they don't have the transporters to take it from outside.
Oh, they always do. Because the auxotrophs, we can feed them with exogenously added things and they will grow. So they will take it up. So that's not a concern. But these two are very good ones, right? So one's about, right, like the dynamics, like if you have delays and it's too late. And then the other one about whether you have even transporters or permeases that allow them to leak out, right? And then what's a third possibility that this could fail? Almost there, almost there. It's related to what you said. Right? If each cell releases very little, right, of course it won't work. It's a trivial case of just the two cheaters being together, right? So all these are very good points. And so that's why I did that experiment first. Going back to the point why I didn't edit it, just so to get a sense about what's going on. It's always easier to go for simpler system, right, before this. Okay, so that's one.
So I missed. This is experimental plot or?
Oh, these experiments.
So, okay.
Yeah. The triplicates, so sorry, I forgot to say, it's like each is like a triplicate, right? Like each color is a different like experiment.
So by looking at it, how do I know it is following first order kinetics? And the model which you made was all linear, right? The mathematical model.
The mathematical model
had all linear terms. In the sense I don't see any square.
So for this one, see, like the point is that it's steady state, it does. And I will show you that. At the beginning, I do not know, because there's a dynamics, because of the delay, I didn't model the delay. Like the initial dynamics, right, as I told Sylvia, right, the point is that the release kinetics is not that straightforward. This is just a very simplified, very simplistic view of what's going on. This is more appropriate for the later stage, right? For the later stage where it grows exponentially. Yeah, so the initial lag phase, we still dare not to tackle that because it's just too messy, right? So it's too messy at this point. So then Babak, he actually came to my lab as a postdoc, and his training was electrical engineering. So he had never actually done any biology experiments. So I just remember when he came, he would just always simulate. So I asked him, why don't you do experiments? Why do you only simulate, right? But anyway, but then later he actually learned to do experiments. He could even do experiments I could not do. It's just amazing how one could develop, right? If one wants to learn it, one does it. So he wants to know how might a spatially structured environment, like the question I posed at the very beginning of my talk, right? Protect heterotypic cooperation. So let's go back to, so now we add the cheaters, right? So we have the cheaters there. And so if we put them on agar plate without adenine or lysine, they're forced to interact in a sense, right? Because they cannot get these metabolites from the medium. So then, of course, they engage in cooperation cheating. So I call it, again, this is focal. This is a partner, and this is a cheater, right? So this is cheating because it does not help the partner, but it grows faster than the red, the overproducing the lysine requiring adenine overproducing cooperative. So then we can also change the environment. So for example, if we supply them with adenine and lysine, then they will not need to interact with each other, right? They only need to get the metabolites from the medium, and we call them competition. So that's sort of like my null, kind of like my background, right? Our background. So what he did is that he mixed the 3 types of cells at 1:1:1 ratio, right? And the vortexed, mix them very well and play them on top of agarose pad without adenine or lysine, and just waited, for like maybe a week. And then he gets to this kind of colonies. And then he will scrape cells off and measure population, ratio using flow cytometry. So in the first experiment, if they just compete, right? We see that so what do you conclude from here? Actually, I should ask you guys, right? So for competition, what does that suggest? So the ratio, right? At the end of the experiment, initially it's 1:1, and then later, what does that suggest? It's not a tricky question at all. What does it suggest? Exactly, very good, right? Because the ratio dropped below, so that means the cheater has advantage, so cooperative has a disadvantage. So adenine overproduction incurs a cost. But if we do this, we get the opposite result. We get cooperators now is favored. So why is that, right? So it turned out so like Babak's spatial model becomes really useful. So he simulated the same process, right? And the way he does it is so like, so I will tell you about model assumptions and the model parameters, how we measure these model parameters. So first is nutrient release and diffusions. So first, release, diffusion. So how do you measure release? How would you measure release? If I tell you this one overproduced some metabolites. How would you so first of all, to release something, you have to measure the concentration of the released metabolites, right? How would you measure the concentration of the released metabolite? I know it's not a fair question because many of you, like maybe physicists, who have never thought about the microbiology, right? But just suppose you do it. Like, what are the? And it's okay if you give like a really, like a, sort of like outrageous answers are welcome too. So, how would you measure metabolite concentration? Very good. How many of these things, cells that needed a metabolite will grow, right? That's very good. So, so that's exactly what I'm. So basically, we take these cells. So if, for example, if I want to measure like lysine concentration, right? So then I take these cells to, of course I have to grow them with the presence of lysine, otherwise they won't grow. Grow them and to exponential phase, so the experiment becomes repeatable, right? Because then talk about complexity of biology, right? Because once cells get into stationary phase, their physiology is very, very different from exponential phase. And why we like exponential phase? Why do experimental biologists like exponential phase? Why do they want to? Of course, you can study stationary phase; it's like a huge field. But for reproducibility, like why do biologists want to take lazy biologists? Why do they like exponential phase? Yeah, that's a very good point, right? Because in exponential phase, nothing is limiting at that point, right? It's just whatever, of course, except for the one that's limiting the growth rate. I mean, otherwise, it's not limiting. So you have a lot of degree of freedom. You can take the OD at the low exponential, mid exponential, right? And they're all the same cell physiology. So then you don't have to get into the lab precisely at that time to catch that moment of cell physiology. Right? In stationary phase, it really differs, right? Whether you're in stationary phase for an hour, for 10 hours, 20 hours, the cell physiology changes all the time. So that makes the experiment much, much harder to reproduce, right? So we catch the exponential phase and we wash them into medium lacking lysine, right? So then we can ask, then we see how many, then we periodically measure. Because so the reason we are doing this is because we want to mimic the cosmos, the community situation. Now, of course, they are actually not having lysine, but this is a kind of approximation. If they are starved for lysine, ideally I want to have to say if they have very little lysine, right? But so for approximation, I would say when they're starved for lysine, and then we measure the adenine concentration, right? And then we can measure adenine concentration in exactly the assay described by the student, right? So I have standards. I know the concentration of adenine because I weigh them out in the scale, right? So I have a concentration, like a series of tubes with known concentrations of adenine. I added these some cells that require adenine to grow, and I look at the OD. So actually it turns out that, in this way, biology is very friendly. It gives me really straight line. So then if I have unknown sample, I can just put the cells into the tester cells into this and then ask what the final OD is, right? And then I just, like look up what it is, and then I know. You can also do like HPLC, like the liquid chromatography, more fancy techniques. Like there are many methods, but this way just fast and cheap. And actually generate results consistent with the chemical ways of measuring. So now we have this, right? So we then also measure live dead cells, because I want to kind of know like whether live cells are releasing or dead cells are releasing, and so on and so forth, right? So now I have this dynamics, right? So these are experimental data. So x-axis is time, y-axis is the live cell density, dead cell density, as you expect, right? The live cell density will decline because these cells don't have lysine and they require lysine. So that is going up, and adenine in the medium is going up. So how would you measure release, from this, the data I collected? So let's think, right? Because you can ask, well, maybe let's think of extreme cases, right? So if only dead cells release, how would you measure it? And then in the other extreme case, if only live cells release, how do you think, how would you measure it? You can give it a try. Yes? Yes. Yeah, that's right. But so then if we consider, right? So I will break that down into two parts, right? So suppose if only dead cells release, right? That's very simple. Like this amount of accumulation is due to this amount of death, right? And of course we plot that, and then we plot the second step, right? That amount is due to that amount of death, the second, right? So then you will see a straight line. But what about the, so suppose I tell you it's only live cells release. How would you do that? Not exactly. Not exactly. Yes, you're getting there, but not exactly. It's different from the dead. Well, what is the? So by the way, dead cells is a femtomole per dead cell, right? So if you look at the units, it's a femtomole per dead cell. It's not a rate, because they die once and then they're done, right? So, yeah. Okay. So how do you measure live cells? Yes, that's right. Integrated over time, right? So basically, it's like the man-hour kind of thing. You have all these people working, and then it's basically like a biologist integral, right? It's like area on the curve, because we don't have that much data, so we still have to do that, right? So for live cells release, this little alpha one is these amount of cells worked through this amount of time, right? So it's the gamma one. And then we do this for the second point, and then we get a straight line, and the unit is femtomole per live cell per hour. Right, so what is it, right? So which is it, right? Like, so if we look at this, so if we assume it's a live cell release, we get a straight line. And if we do dead release, it doesn't look, right, because there's a point here, it just doesn't look that good a fit. So we conclude it's a live cell release. But there's a different way of doing that. You can say for example, this one, right? We say each cell, like because we can take cells and crack them open and then measure how much lysine is in each. Right? And so we know each cell has about 0.12 femtomole of lysine. And we can count how many dead cells is in the medium. So we know it's about 10 to the 5th dead cells per mil. So we expect to see it's 0.012 micromolar in the medium. But in reality, we see 10 micromolar. So we know it cannot be solely due to dead release. So it be vast majority is live release. So that's another way of measuring. But regardless, this is that's how we measure the release rate. So the diffusion, we can just put the dye in here to measure release coefficient across the agar plate, or this way, right? Or this way, so that to measure diffusion coefficient across the colony. Turns out they're different, but turns out like afterwards we try to use the same diffusion coefficient. It doesn't really change any conclusions. So we didn't actually need to do that many experiments, right? So that's another advantage of modeling. Once you know like, which parameters are sensitive, you can save yourself experiments, right? So doing fewer experiments is always good, right? Because experiments are very expensive. Okay, then consumption, we just did the consumption, right? So like it goes down, and then cell division and the cell death. So how do we measure that? So consumption, I just, we did this already, right? So like the earlier graph of the standard curve, we already do. So for cell division and the death, so we can, so for example, again, take this red cells, we grow them exponentially because we like exponential phase. We wash away the lysine, and then we take the cells and put it in the different concentrations of lysine in 96 well plates. We then can do microscopy. So what happens is that in the wells, the cells will divide, and so actually, the total fluorescence actually scales linearly with biomass up to 10 cell layers. So it doesn't matter that so this is actually a very forgiving experiment in the sense you don't have to be in focus. In fact, once it's more than a couple layers, you cannot possibly be in focus for all layers, it's just not possible. Right? But somehow the collected, right, the total photons captured by the camera is linear with respect to the amount of cells in there. So that is a very convenient assay. So we look at total fluorescence against time, and it's a log scale on the y-axis. So like from here, it's a death rate, and from so it's 0, and at 10 looks like this. So it's a net growth rate, right? It's a birth minus death gives you this. So then we can plot, we get something like this, right? So the net growth rate, it's negative, they're dying if you have 0. And eventually it becomes, saturated. Eventually, like around 5 micromolars, they grow at the maximum growth rate. Any critiques of this assay? Dead cells, they don't contribute to the biomass, right? Because they're not flowing. Because once they die, somehow the protein just, pops out, and then they don't register fluorescence. So that's, yeah. So I'm not using death dye here. Yeah, what's your critique of this? The problem is following, right? For this, if you have like a small amount, right, like not 10, like say intermediate level, like say 1 micromolar, it becomes really tricky because as they grow, they're depleting the metabolite concentration. So even though you initially put in 1 micromolar, you don't know at which point it's not 1 micromolar anymore, right? So that's a problem. But for this reason, we add very few cells. So even if they divide a few times, they would, like, we only stop. We don't use data that is near, like say, we only use 1/4 to the maximum. So then we reason there's still 2 more generations to go. So then we're still very close to that number. And also we can calibrate this against, against other means. So we know these values are, honest values, right? So consumption, as we said, right? Like we did this before, like this, how we did the standard curve. And if we 1/over slope, will gives you this femtomole consumed per new cell. Right, because the slope is a cell per adenine femtomole, and so I have to 1/over that. It gets femtomole per cell. So it's exactly the same. All right, so cell rearrangement, right? But it's a 3D, how do we measure that? So this is an image, right? So if starting with a single, like budding cell, and then they grow into this mini colony, right? Oh, okay. Very good question. That's one way of measuring it. So when you grow them, right, you can look at how the decline, how the initial metabolite, you added like it's L minus, right, or A minus. You assay how your initially added adenine is declining with time. And at the same time, how high, the cell density to measure how cell density is going up. And then, and then quantify. Then you can just assume like every single cell consumes something. You can do this that way. The other ways you can do, you can just let them grow to such, to where they cannot grow, right? Because you are not adding enough. So the glucose and so forth, they're not limiting. So eventually the cell density is capped by how much adenine is in the medium. And so for here, I can vary the amount of adenine added, and the density will go to different levels. So from the slope, I can calculate, right? So slope is like density divided by cell density divided by nutrient concentration. But if I do inverse of it, then it becomes the femtomole, because per volume cancels out, right? So molar, actually molar is a mole per liter, right? So the volume cancels out. So you eventually get femtomole per cell. How many femtomoles required per cell? It's about 5-ish, right? So all right, very good. So then at this point, at the 20 microns, approximately, you start to see bright spots. The bright spots are because cells are budding to the next level. So now you have two cells, on top of each other, and somehow they, like it becomes brighter. And then we call this confinement neighborhood. It means that they of course prefer just, be in touch with the agarose because that has glucose and so on and so forth. But when they really cannot push each other around anymore, they just go to the upper level. And then, and you can see the edge will still spread in the monolayer, but the center is getting more and more bright, right? So then top view, so Babak made this model, right? So suppose the star is a budding cell, and it will try to, so this confinement neighborhood. So when it's ready to give birth to the next cell after it consumed whatever it needs to consume, it will look in the confinement neighborhood. Is it empty? Are there empty slots? If the answer is yes, it will try to push this into the shortest path to fill the slot. And if it's completely filled, right? So then it will just push cells up. And if this is a cell, then it just, if it happens to this way, it will just fill. It's a kind of artificial, like sort of an artificial way of describing it, but that kind of captures like the main features of how we think it's working. And other physicists have done using diffusion and so on and so forth. And it's like, I think their patterns are more realistic. So then you have this, so then he used these like sort of like 2-scale model, right? Because a different spatial scale and time scale, right? So what does that mean, right? So like in the cell is like 5 micron, right? But diffusion is much faster than that, right? So then if you always have the the spatial scale very, very tiny, then the computation becomes really slow. But if you have like scale all very large, that becomes really imprecise, right? So it's like sort of like, so he had to juggle between this. So that's a kind of trade-off, right? So basically cell birth death rearrangement, it's a 5 micron. And it is slow, right? Because cell birth death is very slow. But nutrient distribution is much bigger, 60 micron grid. And update is, it's much faster. Diffusion is much faster than birth and death. So then from this, so then he could, because for simulation, the advantage of simulation is that you have exactly the data, all intermediate steps, right? Unlike experimentalists who just sample whatever they can sample. You always wish, oh, what if I had sampled that? But it's too late by the moment experiments are finished, right? So for simulations, don't have this problem. So, but he did it both experiments and the simulations, right? So he would look at the 2 views, one's top view from top, and the other cross section. So actually, it turns out for yeast, it's very hard to do cross section. So because somehow it's very opaque. So the light, you cannot see beyond the top 1 or 2 layers. So then what he did is he froze those, right? He froze those, almost make them like a brain, right? Because the brain people, they slice the brains. So he just freeze them and then slice these, right? Slice these communities and then put on the microscope to see. So let's start with competition again. So the medium has adenine and lysine, so they don't require each other. So these are kind of hard to see, but you can see like the red, they are like, so simulation on the left, experiment on the right, and it's the top view. And you can see it's like very homogeneous, right? It's like a mixture, random mixture. And you continue, it just looks more or less the same. And if you look at cross sections, you see columns, right? You see green, red, and blue columns. And then for cooperation cheating, you start initial conditions very similar. And later you see this, right? So later you see this. So it's actually very striking. Both, although there are differences in the scale, the feature scales, but more or less the green and the red, the cooperator and its partner, its green partner, are well mixed, and the blue cheaters are spatially segregated. Right, and then if you look at the vertical cross sections, you again see the intermix between the red and green cooperators, and the exclusion, the physical isolation of cheaters. And because cheaters are isolated, they cannot really grow tall, so they are at the foothill. So it's almost like they can recognize partner. Almost seems like, although you know they cannot possibly recognize each other, right? Almost seems like they can recognize each other, right? Like it gives appearance that they preferentially interact with each other and exclude cheaters. And how do you think this happens? Anybody who has not spoken, do you want to try? I mean, it's fine. Like mistakes, right? I keep telling my students, it's fine to make mistakes, right? You're not graded, and just it's like fun experience, right? To say something wrong and then to see, oh, that should be that way. Does somebody else want to try? Like, just make some wild guess why that's the case. Is it completely expected? Because the data, like, it's actually not obvious. So why would it be like this, right? So anybody wants to take a guess?
Selective, after biting, if selective migration is possible, I'm not sure.
Selective what?
Like it migrates.
Oh my God, no, they cannot move. They don't have chemotaxis. But they move in the sense that when they divide, right? Like if I'm divided, if if this chair divides into that chair, so of course that chair gets born next to it. So in that sense, the front is moving, right? Like a sort of like they are expanding spatially or upwards, right? When they go to the next layer. So otherwise, they are not activate, they're not motile, those cells. But that's a very good point. In fact, that was one use of simulations, right? So we thought the same. So then we can simulate exactly the same thing, but spatially array them perfectly. So there's no heterogeneity in this initial distribution. The same result holds. It's weaker than, it's becoming weaker now. So spatial heterogeneity definitely played a role, but it's not absolutely required. So I can tell you. So that's another use of model, right? Because you can, of course, use inkjet printer to print those cells onto the agar, right? It's just really hard to do such experiments. So before I go on, I just want to say quantify. You can quantify that, right? Like quantify that. So you can look at the red and count how many, cooperator partners, and you look at blue, count how many the green partners are there, right? So you do the ratio, and you can indeed see, like if it's one equal partner and greater than one as a cooperator has more association with partner than cheater does. And we can see in competition, the equal association. But the minute you have cooperation, cheating, you have higher, right? Like the preferential association with the partner, with green partner for the cooperator than cheaters. So it's a self-organization, I call it, right? So generation of non-random patterns from initially random spatial distribution, right? Solely due to internal interactions. And what is required? So that gets to your question. Like to ask what causes, then you ask what's required for it, right? So what's required? So for example, so what's required for self-organization? Okay, so if we cram them into a little space, right, and allow them to grow, what happens is that in the center, right? So we are looking at this. So let this grow up, and we section at various distances toward the center and see what happens. So expanding front and what happens, right? So we do this. So initially it's a very dense, so they don't have much difference from one. But as it grows outwards, there's a space, right? They can self-organize. Then you have this, like a preferential association between the cooperator with partner compared to cheater with a partner. And so experiments, it's even more striking, right? In that sense, right? So that's how I intuit. So that basically tells me space is important, right? That tells me, so that the earlier experiment tells me space is important. It had to kind of get into open space to do that. And so the way I envision of this is just my hypothesis, right? So if you suppose you have the green partner surrounded on one side by cooperator, the other side by cheaters. So what will happen that, imagine these cells, right? So the cell, the red cell that's closest to the partner will divide more than the cell that's farther away from that partner, right? And when, so that shows the probability. So as it grows, so the cell just bud randomly. But if I happen to bud toward partner, I'll be more likely to bud again compared to if I had happened to bud away from the partner. And so that would cause this piling over of the red over green. So that's open space as well. Right? And of course, the cheaters will do exactly the same. So cheaters will start piling over on that. But now there's a difference, right? So the cooperator side, the partner would have, opportunity to grow either ways. And if they happen to grow on top of the cooperators, it will have more chance to do it again, right? But so the cheaters, nothing happens because they're not favored, or they cannot even divide because they're not getting metabolites. So then this kind of intermixing happens again. So what causes? That's just my intuition from the simulations. So so going back to the beginning of the talk, right? So we mentioned the recognition and the spatial structural environment. Oh, because the cheaters, right? Because there's nothing. So at some point, the partners here are dying off because they're not getting anything back from there, right? So they're not growing, and then it just so they seem isolated. It's just because the cells underneath probably not giving them. They're not getting anything back, right? And the thing from there are preferentially used by those partners. So those partners basically are, right, like sort of like dying. And so cheaters have nobody to cheat from, so they're isolated, right? Eventually, they will just be really isolated and can't grow very much. Or any questions at this point? So going back to the beginning of the talk, right? So we have the recognition, spatially structured environment, right? And recognition is engineered, so it's not there. A spatially structured environment, we understood, right? Like it does, but we can destroy it as well, right? By doing this very well mixed environment. Then what would you expect to see? Very good, right? According to that defect that the cooperator defect that I showed earlier, right? So very good. Okay, so let's see what happens. So adding weight to the experiment. Will cheaters destroy cooperation in a well-mixed environment, right? So of course it will, but we just want to see whether it indeed does. So again, so cheaters have a 2% fitness advantage over cooperators in this case. So then we mix the 3 at the 1:1:1 ratio in a like shaken flask, right? And it have replicates, of course. All experiments need to have replicates. And then we well mixed, and then we grow them to modest density, dilute into fresh medium without adenine lysine. And then we do this again, right? And then, because of 2% fitness advantage, we would expect cheater to cooperator ratio doubles, should double around every 50 generations, right? So then we should expect eventually cooperation would be destroyed. So we have we made a master mix, and then we have 12 communities from same master mix. So they have exactly from the big flask. We inoculated 12 tubes, and then we see how they grow. So the y-axis is experiments. So y-axis is community generations, number of generations of OD, right? The turbidity, the total biomass has done. And the x-axis is time, right? So, so basically if the straight line, that means, so if I see a straight line, what does that mean? What is what? What is y-axis community generation? So like if I started with OD of 0.1, right? Then OD 0.2 is 1 generation. OD 0.4 is another generation. OD 0.8 is third generation, right? So it's OD, like community, like a turbidity, measured by turbidity. Because I, don't really know the ratio of the cooperator children in this. I just measure total turbidity. And then it grow and then I dilute it, right? And then grow up again. So these are generation, right? So if it's a straight line, if community generation is a straight line of time, what does that mean? Yeah, it's a constant, right? So it's a constant. So like, let's say if every hour, if every like 10 hours is a generation, right? So then it would show, then of course 20 hours be 2 generations, so on and so forth, right? So that's very good. And so ratio, but at the 50 generations, we said we expected the ratio, cheater to cooperator ratio to be 2 because of the 2% advantage, right? And we look at this, the cheater or to cooperator is actually greater than 100. So these are really, really vicious cheaters, right? They quickly extinguished the cooperators. And then in some other like replicates, in some other tubes, we saw the opposite. So in those tubes, they're straight lines, so like it means they're dividing in at constant rate. The cooperator to cheater ratio exceeded 20, much higher than the, like it's not like cheater dominating, it's a cooperator dominating by a huge margin. And then if we look at this, across these replicated communities, we can see there are 2 groups, right? So the orange group is fast growing and the gray group is slow growing. And we can quantify the growth rate. It's really bimodal. So then what could be happening here? Why would I see like such distinct, outcomes, right? Given that they form same master mix. Anybody who has not spoken? Any student who has not spoken? Some wild guess, it's fine. It's not an easy problem, right? Because I remember Adam had a thesis committee, right? He showed this result, and the committee members say, oh, you must have done something wrong, do it again, right? So that's what so it's not obvious. Like when I ask, what do you think it happened? So like they say, oh, just like, you messed up something. Right? But he said, no, but that's from same master mix. He said, doesn't matter, just do it again, right? Because this is just so weird. He did again exactly the same, more or less same results, right? So what do you think could have happened? If you were Adam, like what hypothesis would you make? It is. You're getting there. Yes, it's a clue. Yes, it is. It's initial to the. It's sensitive to the initial like ratio. If I do so, the hint. If I do initially 10 cooperator to 1 cheater to partner, this will be 10 orange and 1 gray. So the fraction of the orange and gray is sensitive to the initial population size of the cooperators and the cheaters. You can ask questions just like what he does, right? Like you can ask, does this do this? Eventually you will know the answer. So I give you a hint already, a very strong hint. It's a very good question. Yes, it does depend on the initial ratio of cooperator to cheater.
How often do you notice it? Like, is it constantly spinning, or is it that once you've...
It's constantly spinning. We have a spinning wheel that doesn't stop, like a 24-hour, right? 24/7 spinning. Sylvia. That's a good question. Let me think. It's about 10 to the 7 cells. That's a good question, yes. Yeah, there are a lot of cells for each strain.
So when you see something come out from orange versus white, do you see any difference in the contents of how it looks, like in terms of...
Oh yeah, it does, right? So like this, the ratio. So for gray one, cheater dominated.
Are they clumped in any way?
No, they're not clumped in any way. But that's a good point too. But you're getting there, right? Because they could evolve somehow, right? Maybe evolution. Because then it will depend on initial population size, right? Because whoever has a higher population size would be more likely to, to win, right? So, so actually, so we, so basically, with the yeast, because and also with the genome sequencing, it becomes so easy just to see what's in the end point, right? So we call this, we do deep sequencing, and you can sequence like say, 70 strands in one Illumina lane. Like a pair, and it's just like revolution. That was a revolution. I remember when I was a graduate student, I would isolate these mutants. It would take me a year to figure out what that is, right? Like I have to clone the genomes into this library and the complementation, see which one complement in the sequence. And I remember like went through the sequencing, because there's a Sagar sequencing, right? Because this isotope labeled radioactive, and like it's a very big plate. I run them, and then I have to pry this open. And I was just praying to God, please, when I open it, please don't crack the gel. Because it's so much work, right? And I crack it and I put it on a piece of paper, like a filter paper, and dry it and expose to X-ray film, right? And I just remember like how many times I prayed to God, please don't let it crack, right? But there, like now, it's just like you do the library, you send it to some company, and after a while, you use a huge amount of data. You don't have to even construct libraries. You just compare the sequence, the mutant sequence against the ancestor sequence, and you will see, oh, these nucleotides have changed. Those are your candidate mutants, right? So deep sequencing, so Adam had his life easy. So they found all these, and also yeast has a very good reference genome. So like each gene, like yeast has about 6,000 genes. Like, some genes are still mysterious. People still don't know what they do, but many genes have been studied. And we have a yeast database talking about what each gene does. So lucky for him, all these mutants are hitting genes with names, right? Names means people kind of know what they do. So what happens is the following, right? So that's how yeast works. So basically it's like it's a cell membrane. It has these permeases on the surface, so they can take up lysine. So there's the one called the lysine permease, called the LIP1. It's a high affinity lysine permease. It has a vacuum. Vacuum is like our stomach, right? So what happens is that in normal cells, because when lysine minus cells get into, so wait, there's no lysine because it's very stressed. So when stress comes, there is a, sorry, like I was too fast. There's some called ubiquitin, it's like ubiquitin ligase, and don't worry about what it is, but it's like some proteins that attach a ubiquitin. Ubiquitin is like a little protein, like a moiety. It attaches a chain of ubiquitin to it. So then that's a signal for cells to eat it. So the cells will then take this up, right? And then this will escort, this will actually recycle, because ubiquitin is precious. It recycles this ubiquitin, and the rest fuses with the vacuum. And it gets degraded, right? And then these amino acid then is liberated into cytoplasm to help, the halted protein synthesis to continue. Right? So you see, like a biologist know a lot of how it works, right? But it's just in this kind of model structure, right? But in this mutant, when you have this inactivated, what happens? So those genes are responsible for degrading like LIP1. So in the mutants, those are like inactivated. So what happens to LIP1? Yeah, there will be more of them. They're stabilized, right? There will be more of them. It'll be unlike they're not degraded, so they are so allow this. So that's why they have high affinity. So that's why they're very good at the taking of lysine. So these cells, so if we look at the LIP1, we can tag it with GFP, right? So the ancestor you can see at the low concentration, they all get into the stomach, right? The central vacuole. But the minute you have this evolved background, they're all on the membrane, right? So stabilized they're getting very good at taking up the very limited amount of lysine from the partner. So then if we measure that net growth rate, we can see, right, that's, the evolved grows faster than ancestor on the low concentration lysine, which is like the cosmal region. It's like around 1 micromolar. Although they have a fitness cost at high concentration of lysine. So like this and that, right? And then, so we have this called sort of like adaptive race between cooperators, cheaters, right? So basically, initially you have like, the cooperators, cheaters, they are, and because of like a Loria-Delbruck kind of phenomenon, some mutations might be pre-existing already, right? Because the mutations happen randomly. So the mutants, right, they are initially rare on the excess lysine because these have fitness cost at the excess lysine, but they are there, just rare. But when you mix them, and the partner is giving them a very little lysine, So then these pools, the cheaters, cooperators, they're basically asking who has the most fit, like a mutant to deal with the slicing limitation. So stochastically, if it's a one-to-one, right, 50% probability, the cheaters would get the highest, most adaptive mutation, and it will, win. 50% it's a cooperator gets the best mutation. And in this case, they will just grow up, they will just crash the whole system. In this case, they might even be able to get rid of the cheaters. Right? So that is, and then you can imagine, right, even though it's a synthetic system, you can imagine, like, so of course this cycle can start again, but again, right? But you can imagine that different type of environmental stresses, like a changing temperature, changing salinity, antibiotics, all this will give you opportunities to, for cooperators to have a chance, not necessarily they will win, but they will have a chance to, to purge cheaters. Right, so now so this class, this lecture, we covered two, mechanisms, right? Spatial self-organizing and stress-induced adaptive rates. And this only became apparent, right? Because we have engineered a system, then we have excluded all known mechanisms, right? So then allows us to see this phenomenon. And people have seen this in natural system as well. And so in both cases, I want to caution, right? Like I want to mention, it's all involves small population size, right? In the sense here, your initial mutants are rare. It's small. If it's large, it's a deterministic, no longer stochastic. In there, it had to be small because if it is it had to be small in the sense that it had to be expanding front or they're going up, right? So it's like, because again, at a large population size, the stochasticity no longer works, right? So that it is important. I think that's a shared feature between the two. So any questions? It's a same class of mutations. So either like, it's actually really amazing, right? Like all these ones, they all, because thanks to yeast literature, we were able to figure out in no time. Once you sequence, you know the answer, right? All this, yeah, they're in the same class. They're all involved in degradation of the lysine permease. So mutations in these would give you the stabilized lysine. So the cells have different ways. They also have different ways, like a duplicate. They can duplicate chromosome. So they double the copy number of LIP1. That's another strategy. Right? So it's more or less the same kind of like a class of mutations. No, but those ones are selected. So those ones are not selected won't show up here. These ones are enriched for those ones are very, very good at competing for lysine from the partner. You mean for the partner? Yeah. Adenine deficient. In the sense that if I start, yes, I would expect the same thing will happen. There's adaptation, right? So the cooperator cheaters, for the for the one that's up, we didn't do the experiment. We suspect the same thing would happen. Oh, then the follow up on Adam's story, right? So once we show this, the biologist said, "Oh, that's trivial." so when they see this is stochastic dynamics, they say, "Oh, do it again, because you must have done something wrong." But once you understand, they say, "Well, that's trivial." So that is that is what But still, I think it's a very good piece of work. So for PhD students, right, the advice is ignore what other people tell you. If they say, "Oh, you must have done something wrong," ignore that. If they say it's trivial, also ignore that. Right? You just do your own science, sort of. Yeah, so I'm asking you, what is a deterministic behavior? That's a very good question. So suppose I started with lots of cells. Or here, I cram the space with tons of cells. What do you think will happen? Yeah, so here, it's a complete cram. There's no space to self-organize. Like I just have this petri dish, right? I just cram it with cells. No, cheaters are there too. That's right, it's a deterministic, right? In the other case, suppose I start not with a million. I start with 10 to the 12 cells, for example. So then guaranteed every culture has about the same amount of lots of these mutants, lots of that mutant, right? So on average, then cooperator cheaters would have very same adaptive mutations now, right? So then the cheater advantage of 2% shows up again, right? So I didn't mention, that's a good point though, because during adaptive race, right, the cooperators had to sample mutation. There's at least 2% more fit than the best cheater mutants. Otherwise, you're not going to win, right? Exactly. So then if you have tons of cells, then, then I would just imagine you average, like you have the best, I have the best, because I have so many big population, you too. But then your best, if you're a cheater, your best will outcompete my best, right? So in that case, it will be deterministic again. But that's thank you for that. I know I didn't elaborate that. I was waiting for, sorry, I was waiting for your question. Yes, I would. I've not done experiment, but that's my prediction, yes. Very good. Oh, yeah, you have a question, yes. Yeah, no, that's a good point. But what I was saying the following, right? If you have tons of cells, like I don't know the number, like say 10 to the 12 cells, right? 10 to 12 of cooperators, 10 to 12 of the cheaters, right? And for each of one of them, then there's such a large population, they can sample like the best mutation, like it's A star. It's able to sample A star because it's so large, maybe it sampled maybe 10 times, for example. And this one maybe sampled 11 times, right? But the cheater, this says this is cheater, this is a cooperator. This cheater, so these are identical otherwise, right? Except that this one cheats. So then these ones, so dynamics basically, instead of the big population, it becomes the competition between the best of the best, right? The best of these adaptive mutants. So if it's a many, so then the number becomes determined. It's always like, so it's not like, so if I start with, in fact, I should do this. If I start with 10 to the 6 cells, right, 10 to 6 cells. So maybe this one happened to have 1 A star and this one has 0, right? Like 1, and this is 0 A star. And even though this can show up later, but I already have advantage at the beginning, right? So then I will take over. And then and as I do dilution, all these, like the chance becomes less and less. And for this one, I start with more, because this population size is so large, I start with similar numbers of this, right? And then it becomes cooperator-cheater games again. So the cheaters would be more fit than cooperators, then they will just, because they're not overproducing metabolites. So they will slowly but deterministically take over, right? So, but it will not, you're right, the gray won't be that quickly dying. You'll be slowly creeping up, right? So the ratio of the, sorry, the ratio, like this is not the right graph, right? So the ratio won't be like, so for adaptive race, because the cheater wins, they, like the cheaters goes, like I would say, the cheaters, the ratio can go up really quickly. So cheater to cooperator ratio. Some of them like this, some of them like this. But if it's like, if in determinist case, it would just go up like the 2% at a very gentle slope, right? Because it's the fittest against the fittest. Yeah. So. Thank you very much. I think, I don't know what's the schedule next. It's a